What we are about to learn might give us a new perspective on existential questions such as, who are we? Surprisingly enough, we should actually think of ourselves as colonies composed of billions of different living things. Indeed, in our bodies, there are as many bacteria as human cells. And the number of bacterial genes inside us is about 100 times the number of human genes. The next time you weigh yourself, consider the fact that at least 1 kilo is due to the microbes living in your gut, called gut microbiota. These tiny inhabitants play a vital role, helping us digest food to obtain energy, producing vitamins, breaking down toxins, and so on. Andrew Macpherson in Bern has devoted his life to the study of this overlooked part of ourselves, and he is currently leading a project funded by Systemsx.ch.
Our bodies are populated by enormous numbers of bacteria. These live in the intestine and on many body surfaces, and often do us absolutely no harm. Depending on which sort of bacteria are present in our bodies, we can be healthy or they can cause disease. So examples of these diseases are diabetes or depression, but many other conditions are also influenced. If we are healthy, then part of our health is due to the exchange of molecules between these different bacteria and the rest of our bodies.
Basically, there are no two people in the world with identical gut microbiota. It develops during childhood according to our environment. And although it will resemble those of our parents or the people who live with us when we are young, it has its own unique personal character.
Not all the bacteria that are present in nature can actually colonize the intestine, although the number present in the intestine is rather large, about potentially 1,000 different species. Of these, each of us has about 100 different species present in our insides.
If the human body were planet Earth, the gut would be the rainforests. Here, thousands of different organisms live harmoniously. The system might look chaotic from the outside, but it is not. At a closer look, each species has its own role, and there are almost no unexploited resources. That is why immigrant species have a difficult job of integrating. But how does our inner rainforest work?
Effectively, we've only just started the exploration of the gut microbiota. As part of the Systemsxch project, we are trying to understand how the exchange of molecules between the different sorts, the different species that exist in the gut microbiota works, and how the exchange of molecules between the different species of gut microbiota and the rest of the body influences our disease or health status.
The gut microbiota is located in the mucus that lines the internal walls of the gut. To study it, researchers collect samples by endoscopy. Then a team of biologists and mathematicians works to develop models that can predict the interaction of the bacteria in the different samples. And their results are compared with what is observed in reality. One important goal of these studies is to help patients with inflammatory intestinal diseases. The number of bacteria species in their gut is too low. The rainforest turns to desert. And just as in natural environments, this drastic change is very difficult to reverse.
Unfortunately, in many clinical situations, it's rather difficult to be able to introduce good bacteria to restore health in a therapy context. The reason for this is probably that it is such a complex situation in the intestine that it's difficult for us to decide exactly which bacteria we should use as therapy for patients. Once we know exactly who our gut inhabitants are and how they relate to each other and to our bodies, we will be able to start to design therapies and to use the clinical situation in order to improve patients' health.